Good morning, everyone. I hope everyone is doing well. Today, we will review our lesson about the humoral immune response. First of all, let's take an overall view about our lesson from the beginning. So the immune response is divided into two responses. We have the non-specific immune response and the specific immune response. In the non-specific, we can call it also innate. What do we mean by innate? It means that it's new at birth. However, the specific, we can call it another name, which is adaptive. Adaptive because it adapts with the environment around it, with the viruses. So the non-specific, it has actually two defense lines. The first defense line and the second defense line. The specific, it has the third defense line. What do we have in the first defense line? We have the natural barriers, like the skin, tears, and mucus. In the second defense line, we have the inflammation reaction. Who is responsible for it? The macrophages and granulocytes. While in the third defense line, we have two immune responses. The humoral immune response, which is involved by the B lymphocytes, and the cell-mediated immune response. The one responsible for it is the T lymphocytes. This is a general review. You have to write this concept map on your copybooks. So these are the three defense lines. As you can see at the first, we have the natural barriers. Here, like the skin, it's a natural barrier. The virus, it prevents the virus from entering. In the second defense line, we have the inflammation. It's not specific. But in the third defense line, the immunity becomes specific and it catches the virus from its antigen. That's why it's specific. It knows which virus it's attacking. So when do we use the specific immune response? So this is the innate immune cells. They act as the garbage man. But sometimes some viruses and bacteria get away and they can get inside our body. So it needs backup. Who is this backup? It is the adaptive immune response. So today we will start with the humoral, as we said. So who produces the antibodies? Which cells produces the antibodies? The lymphocytes, and precisely the B lymphocytes. What do these antibodies do? They actually just bind to the antigen and catches the antigens of the viruses and bacteria and prevent them from moving anymore. That's it. So while it is preventing it to move, it is making agglutination. Agglutination. This the process where the antibodies bind to the antigens of the virus and prevent it from moving anymore and makes clumps because they are all now together. It's called agglutination. So where will we study this response, the humoral immune response? In our blood groups. So mainly we have 4 blood groups, as you know. The AB, O, A, and B. You have this photo on your books. They call the antibodies and antigens. Here we have the antigens, they call them agglutinogens. Why? Because they make agglutination. Also, the antibodies, they call them agglutinins. So when we have an agglutinogen, which is the molecule on our red blood cell, is A, our blood group is A, but we do not have the similar antibody because we don't attack our blood groups, never. We attack the other blood group, so we have anti-B. Similarly for B, if we have antigen B, then we will have antibody A that will attack A, not our own blood group. When we have a blood group AB, we have both antigens, A and B, and we will have no antibodies, we will have no agglutinins. However, O will have no antigens, but will have both antibodies. So please also write this on your copybooks, the definitions of agglutinogens and agglutinins. Agglutinogens are the antigens, agglutinins are the antibodies. Agglutination is the process of clumping of the cells or particles in the fluid medium. This is the agglutination. The red blood cells agglutinate with the antibodies and they clump together in the fluid. This is the photo on your books. Now here we can see the agglutination. So when we put antibody of the same type, like for example here blood group A, if we put antibody A, so this is the agglutination that happens. They will agglutinate. So now let's take an example, just to understand. If a donor that will give me blood, I want to see its blood if I will accept it or reject it. So this is the blood of the donor. The donor has type A. My antigens, my antibodies, or my blood is type A. So if I am type A, I will have antibodies B. A and B will not bind together. If they do not bind, they will not make agglutination, no agglutination. So the blood group will be accepted. So if you look at it, this is A, blood group A, and this is also blood group A that has antibody B, so it will be accepted. However, in this case, we have A, but the recipient has type B. So if it's type B, it has which antibody? It has antibody A. So A and A, they will make binding. Antigen A and antibody A, they will make binding. Binding, thus they make agglutination. And agglutination means the blood group will be rejected. So there is another factor we need to know about. We said that we have 4 main blood groups, but we also have another factor, which is the RH factor, which is another antigen like the A and B. We call it rhesus, RH. So when we have RH, we call our blood groups with positive, like AB positive. Positive means it has the RH factor. If it doesn't have the RH factor, then we call it AB negative, and so on. That's why now we have 8 blood groups with the positive and negative, whether we have the presence of the RH factor or the absence. Now, how do we determine our blood groups? We have this test in the lab. We have these 3 are antibodies. So if they make antibodies, if these antibodies make agglutination with our blood, then we have its antigen. Let's take this example. Here we have a blood group. Here we dropped anti-A, anti-B, and anti-D. When we say anti-D, it means for the RH factor. This one did not make agglutination, so it doesn't have antigen A. This blood group doesn't contain the antigen A. This one made agglutination, so anti-B made agglutination with the blood, so this blood contains antigen B. And here it made agglutination with anti-D, so it contains the rhesus factor, so it's positive. That's how we knew that the blood group is B positive. Here you can know all of the results, no matter what is it. So for example, when it makes agglutination with 3 antibodies, then we have anti-A, then we have antigen A, and antigen B, and antigen rhesus. So it is AB positive. However, for O negative, it doesn't make agglutination with any one of them. So it has neither antigen A, nor antigen B, nor antigen D, or the rhesus antigen. So it is O negative. Also, we need to know that not all blood groups can give blood or donate blood to all blood groups. For example, here you can see that the O negative can donate blood to everyone, to all the blood groups. However, it can only accept blood from itself. Vice versa for the AB positive. The AB positive actually accepts blood from all the blood groups, but it doesn't give. Here it is. If it's donating, it cannot donate to any blood group except itself. Also, one more thing, that the positive blood groups only can donate to the positive ones. However, the negative can donate to the positive and the negative. So here is an overall for how the blood groups works and how who can receive blood from whom. In this map you can see, and it can be clarified for you which blood groups can donate. So if you can see here, the O can give all blood groups and itself. However, the B can give no blood groups, but it can receive from all the blood groups. And each one of these blood groups can receive from itself. Because AB accepts from everyone, we can call it a universal recipient. However, the O, it gives everyone, so we call it universal donor because it donates for everyone. This is our lesson for today. I hope it was clear for you, and good luck.